I'm Dr Patrick Baker, Director of Studies in Molecular Biology and Biotechnology. In my research, one of the threads of the research is looking at toxins from pathogenic bacteria. And I want to understand how those toxins are exported from the bacterial cells and how they interact with the host cell, which often in the bacterial world is actually other bacteria. But of course, if the bacteria are infecting a human, then the host is the human and has unfortunate medical side effects. To do that, I'm using the technique of structural biology and in particular X-ray crystallography, where individual protein components are studied under intense X-ray beams to discover the three-dimensional structure, the position of all the atoms in those protein molecules. And lots of bacterial toxins are protein molecules, they're protein toxins. I've been looking at pore-forming toxins, so these are proteins that bacteria export, assemble on the host cell membrane. They just dig a large hole in the membrane, and then the cell contents can leak out and the bacteria can use those as nutrients. Now there's a really rather interesting trick that the bacteria play, because they have to be able to export those proteins through their own cell membrane without destroying it. It appears that the bacteria produce a protein molecule that's inactive, and when it's exported from the cell, it undergoes a very large conformational change. It inserts into the cell membrane. Often 2 or 3 components come together to form a large complex with many protein molecules in it that then puncture the cell membrane and act as an active toxin. Now, understanding that process, that transition from soluble to attacking form, it's really important to know exactly what the three-dimensional structures of all the different types are, and therefore to understand how the toxin molecule changes its shape, transforms from one to the other to become the virulent form. And the role of all this, so looking at bacterial protein toxins, is that if you can understand their three-dimensional structure and understand their assembly and the molecular interactions they make, then small molecules can be developed to stop those processes happening. For the case of the pore-forming toxins, you could develop an inhibitor molecule that would stop the assembly on the membrane. You've then targeted the virulence of the pathogenic organism. And if you can stop the virulence, i.e. stop the bad things happening to the host cell, then conventional antibiotics, which as we know are losing their potency, have a much greater time span to act to clear the bacteria from the host cell.